Hi, I'm Susanna with Science with Susanna, and tonight we're going to study the nervous tissue. So here we go, Introduction to Nervous Tissue. We'll start with neuron structure. Neurons have dendrites. Dendrites help to receive input from all the nearby neurons. Then there's the cell body. I'm coloring this in yellow. It's where you find the nucleus, which is the most metabolically active part of the neuron. This is because it's making so much protein. It makes this protein with structures called Nissl bodies. These are basically just a fancy name for the rough endoplasmic reticulum that you learn about in other cells, but it was named because it stands out so clearly in neurons. And a scientist with the last name Nissl is the one that described this. You can see it in this histological sample that I found here. This dark purple indicates massive protein synthesis. And the proteins are all going to be enzymes that are critical in the development, maintenance, and transport of all the different neurotransmitters that a neuron can make. Neurofibrils I'm drawing next in black. These are cytoskeletal elements that give the neuron its shape, like all of the different processes that come off of it, whether they're dendrites or axons, etc. So it gives it its shape and also all that transport. You see this really long axon and things have to be transported up and down that. Next, I'm coloring the axon hillock. I'm making this blue. This is where an action potential will be generated or initiated. And it's the most common site that we're going to see for action potential generation. Next is the biggest and most prominent looking part of a typical neuron. That's the axon. It's the conducting region of the neuron. It is usually encased and wrapped up in a bunch of fat called myelin, and this myelin insulates the axon with the purpose of speeding up the conduction of the action potential. This fatty covering insulates the axon in order to increase the conduction speed of an action potential. The nodes of Ranvier, I'm putting little purple stars on. These are gaps in the myelin sheath that allow the propagation of the action potential to occur. The action potential starts at the axon hillock and then it jumps from node of Ranvier to node of Ranvier all the way to the end of the axon. The ends of the axons are called the axon terminals, and they're going to be the business end. They're transmitting signals to a target cell. Usually we're talking it would be another neuron. It could also be a muscle or a gland. So these communicate with the target cells by secreting neurotransmitters. Then we have occasionally something called a collateral that I've drawn in light blue here. This is a branch off of the main axon. Now, if we're going to think about the direction of the electrical signal as it goes through a neuron, it comes in through the dendrites, passes through the cell body, goes down the axon, gets to the axon terminals, and then it's passed along to the next target. Okay, so here we're going to talk about the types of neurons. The first kind I'm going to talk about are multipolar neurons. Then there are bipolar neurons, and there are the pseudo-unipolar, or sometimes it's called a general sensory neuron. First, let's look at the multipolar neuron. So, like you're used to thinking of, a dendrite is going to receive the signal, right? And then the axon hillock will generate the action potential, and then the signal will go down the axon. But here's where a multipolar neuron really stands out and gets its name. Do you see how it has multiple dendrites? So multiple dendrites, you can remember multipolar. Then we'll look at the bipolar neurons. Like we saw with the multipolar, the signal's received through the dendrite, but it just has the one dendrite, and then it has the one main axon. This is very common in like eyes and stuff. Okay, then over here I have the pseudo-unipolar, or the general sensory nerve, and it is a little unique in that it has these receptors And then the axon hillock of sorts, if you could call it that, is right there. That's where the action potential gets generated right away. And so they try to call the whole thing just one axon. But you can see that there is a structural difference, right, between the bipolar and the pseudo-unipolar. So you're going to have one main axon. Now, the bipolar and the pseudo-unipolar, they can be remembered because they're always associated with sensory information, whereas the multipolar can actually be both. But the multipolar is going to transmit most motor output signals. It's the most common type of neuron in your body. Compare that with the bipolar neurons that are very specific in sending sense of sight, taste, and smell to the brain. The bipolar neuron is only going to have one dendrite and one axon, whereas multipolar has multiple dendrites and one main axon, and the pseudo-unipolar has no dendrites and just one axon. So the multipolar is your typical looking neuron that if you're taking an A&P class, that's what they have you look at when you're studying histology and like, oh, here's nervous tissue. Here's a multipolar neuron. Next, we're going to talk about the different kinds of neuroglial cells. The word neuroglial means nerve glue, and these are cells that maintain the health and function of the neurons. So the first kind I'm going to show you is called an oligodendrocyte. These funny looking orange cells that I'm coloring in right here, they wrap the axons of the neurons in the central nervous system with myelin. They're able to myelinate multiple axons at one time within the brain and spinal cord. When axons are myelinated in the body, they appear as white when you look at them with histology. So we often just talk about that with white matter. And if you see white matter as opposed to gray matter, that's telling you the white matter is busy sending the information as quickly as possible. Maybe like a factory would be the gray matter, and then the freeways that get the substances from one factory to another would be the white matter. So myelination indicates to you speed. In the disease called multiple sclerosis, it's an autoimmune disease where you have a demyelination of these demyelinating disease where the oligodendrites are not able to keep the axons myelinated like they should. Okay, now I'm drawing another neuron over here, and I'm showing you another neuroglial cell. Neuroglial cells, whether it's oligodendrocyte, Schwann cells, or the other ones I'm going to tell you about, they don't send electrical signals like neurons. They're there to take care of the neurons. I tend to think of it as like the neurons are the trees and the plants in a garden, and the neuroglial cells are the caretakers of the garden. So Schwann cells have the same job as oligodendrocytes, but they are only able to myelinate individual axons at one time, and they're found in the peripheral nervous system rather than the central nervous system. Once again, though, they're going to appear white, the axons that are myelinated. And a key concept here, why do we care about all this myelination? It increases the speed of conduction of the action potential. There's another disease called Guillain-Barré disease. This is also an autoimmune disease, and it's when you have a demyelinating disease of the Schwann cells. So in this disease, you're going to have symptoms first in the peripheral nervous system, like making muscles move. And in multiple sclerosis, they can have central nervous system symptoms as well, sort of like fatigue and brain fog. Number 3 are the astrocytes. These are the most numerous kind of neuroglial cell, and that word astro means star, so these are star cells. Their job is to facilitate neuronal communication. So I'm going to draw you here an astrocyte. It's the one I'm coloring in green, and these astrocytes, do you see how they are attaching to where the synapses are? And that's how they help facilitate neuronal communication. Now this next thing I'm drawing, I've drawn a blood vessel, and I'm trying to draw your attention to how the astrocyte puts its little pseudopods onto a blood vessel in order to block substances from coming into the brain that could hurt it. And we call that the blood-brain barrier. Now, astrocytes, they divide really fast. So here I'm drawing like mitosis of lots of astrocytes. Because of that, they're the cell type that's most implicated in brain cancer. Next are the microglial cells. These are named the small glue. They're the tiniest of all of the neuroglial cells. And their job is to act like the immune cells of the brain. And when they get activated, they become phagocytic, meaning they can engulf debris. And when there is an injury, they're going to migrate toward it. So you're going to find them concentrated in areas of the brain that are experiencing injury or inflammation. So here I'm drawing that they are concentrating maybe in an area where there's been inflammation or damage. I guess I drew a lot of them. Next, the ependymal cells. These cells are the cutest of all the neuroglial cells, and you'll notice that they have cilia. Those are those blue squiggly lines that I've drawn on them, and they do the wave like this, and they move cerebral spinal fluid around in the spaces within your brain and then all the way down your spinal cord too. So they line what are called the fluid-filled ventricles of the brain. Now those are the main neuroglial cells. So to show you this, I'm going to draw just a really typical cell. And the negativity is because of a very active channel. It's a pump called the sodium-potassium ATPase channel or pump. And what it does is it uses one ATP molecule and it will pump 3 sodium ions out of a cell at the same time that it is pumping 2 potassium into the cell. So you can see more positive ions are going out than positive ions are coming in, which gives you this relatively negative environment inside of the cell. So the results of the pump are that sodium is always more concentrated outside of the cell. And that means whether it's just like right outside of the cell or what matters clinically, it's going to be in the serum or the plasma of the blood. And then compare that with potassium that is always more concentrated inside of the cell. But in a clinical setting, we usually don't talk about what the concentration is inside of the cells. Where they're measuring it, like when they test your blood, is how much potassium is in your blood. And you'll see that there's only about 4 milliequivalents per liter of potassium in your blood. That's a huge difference, right? So lots more sodium outside the cell, lots more potassium that's sequestered inside of cells. So the summary statement is that the resting membrane potential of a cell is relatively negative, about minus 70 millivolts. What I mean by that again is that there's positive ions outside the cell and there's positive ions inside the cell, but there's so many more outside of the cell that compared to each other, the inside has a negative membrane potential. And that's just the way our cells are designed, and it makes them ready to jump into action, like an action potential, if you could get a moment in time where there's more positivity coming into the cell, it's going to make something happen. So I'm drawing a neuron here. A little bit of myelin sheath on there. And here is the axon hillock. The signal or the graded potential is what we'll call it, and I'll talk about those more in a little bit. It will arrive at the axon hillock, and if the signal is strong enough, then what will happen is an action potential can be fired. So at this place, there's a bunch of sodium channels. And since the sodium is always more concentrated outside of the cell in what we call the extracellular fluid, then if a sodium channel opens, sodium will always rush into the cell. It's like, woohoo, time to play. And it goes from where it's crowded outside of the cell to where it's not crowded inside of the cell. So we say there's an influx of sodium if a sodium channel opens. The same pattern is seen, but in opposite with potassium, which is always more concentrated inside the cell in what we call the intracellular fluid. So in this case, I'm putting a potassium channel on here in green. Potassium is more concentrated inside of the cell. And then what will happen is if one of those potassium channels opens, potassium is going to leave the cell. And we call that efflux. So influx for the sodium coming in and efflux for the potassium going out. Essentially, this is what's going to happen during an action potential. You're going to get a bunch of sodium that's allowed to come in just for a moment, and then some potassium that gets to go out just for a moment, and that's an action potential. So then you have to restore everything though, right? Sort of like after kids have been playing and there's stuff all over the floor, all those sodium ions that came in need to be pumped back out again so that we can go back to that negative membrane potential. So sodium always stays more concentrated in the extracellular fluid. And then the potassium, which I'm coloring here in green on the sodium potassium ATPase pump, is going to be pumped back inside. So potassium is going to remain always more concentrated in the intracellular fluid. And don't forget that this is an energy hungry kind of process. So it uses ATP in order to pump that sodium out and in order to pump that potassium in. Okay, now I'm just drawing you a little bit more of the axon. Do you remember I told you that the myelin sheath is there to insulate the axon? So in that place, there's not going to be any need for depolarization or letting the sodium in. So the signal is going to jump, and then the sodium will get to come in, the potassium will get to come out, and the sodium-potassium ATPase pump will clean everything back up again and make sure the sodium gets pumped back out, the potassium gets pumped back in again. Essentially, I'm showing you in this tiny drawing here what happens with an action potential. It gets initiated on the axon hillock, and then it gets propagated after each myelin sheath piece. So here I'm showing the potassium getting pumped back in to set things right again. And then a little bit more myelin, and then it would be propagated again after that piece of myelin. What I'm drawing up here in this top left corner is kind of just a quick view of what the neuron parts that we're going to care about for this are. So don't forget these terms. You've got the axon hillock is where you're going to have this concentration of sodium and potassium channels. And then we're going to look at that more closely right here. Okay, so I'm drawing a sodium channel with that little line tells you, oh, it's closed. Sodium's not allowed to come in yet. And then potassium has a closed channel right here. So we're at that negative resting state called at minus 70 millivolts. And I'll use these purple negative lines to remind you it's relatively negative inside of the cell. At this time, both the sodium and the potassium voltage-gated channels are closed. When I say voltage-gated, I just mean that what determines whether they open or not is what the membrane potential is. So at this negative state, they're both going to be closed. Now here I'm drawing a graph, this is classic, of an action potential. And this purple line is what it looks like at the resting state. But you'll notice I made a little bump, didn't I? And that shows you that if you can get a little bit of positive ion coming in, then perhaps the axon hillock can reach what's called threshold, which is usually about minus 55 millivolts for a neuron. So if this happens, look at my picture now. The voltage-gated sodium channels will open, and sodium will be allowed to rush in. And look, now we get what's called a reversal or a depolarization, and it's all caused by sodium entry. And woo, look at that blue line going up. It can go up to about plus 30, sometimes plus 40. I did draw it at plus 40 in this picture, but anyway, you get the idea. It's around about there. So you're going to get this spike is what we call depolarization. Then those voltage-gated sodium channels will close once it gets to this positive point. They don't like to stay open anymore. They kind of like swing shut, actually. And now I'm drawing, again, the sodium channels. They're going to close. And the voltage-gated potassium channels are at this point now fully open, and potassium will leave the axon. So you're going to see, let's see, so if potassium goes out, you're getting less positive inside. So you're going to have a return to the negative membrane potential, and we call this repolarization. And even, do you see how it even goes down a little bit lower? We call that hyperpolarization. Like if someone is hyper something, they're like extra that, right? So we get to this extra negative point. And now we're going to have to close all the channels and restore things back to normal with that pump, right? So in this last picture, it's going to look a lot like our first picture, where now the sodium channels and the potassium channels are closed. We're back at this negative resting membrane potential. So do you see how it looks like our first picture, right? The sodium and potassium channel pumps will pump the sodium back out and the potassium back in, and this will reset the membrane so it's ready to fire again. So you can see in my drawing, I drew purple going right back up to where we started. And that, my friends, is an action potential. So you have resting membrane potential at -70. Then you have a stimulus that if it's strong enough and it can get to -55, it will open the voltage-gated sodium channels and you get depolarization. The cell briefly goes to all positive, and then it peaks out, those sodium channels slam shut, and the potassium channels are open at this point, and so potassium is able to leave. And the potassium channels, their doors don't close quite as fast, and so extra potassium can leave the cell, and you get this hyperpolarization. That's actually a really protective thing for the cell because it makes it so that axon hillock cannot respond to another stimulus momentarily. We're talking in milliseconds here, but you have a resting period. So they call it a refractory period. Absolute refractory period means no strength of stimulus is going to initiate another action potential. And then there's the relative refractory period, which is getting it's kind of coming back up again, and at that point a very strong stimulus could get another action potential, get those sodium channels open again, and then zoom, you're back up again. Okay, so once it's been initiated, we've got to propagate it. An action potential, with another fancy term for an electrical signal, is initiated at the axon hillock. And I've drawn a little one for you there. And then they must be propagated repeatedly until they reach the axon terminal. So I'll show you here with a neuron, those are some dendrites, the cell body, and then here is like the money market, right? The axon hillock, and then you're going to have the myelin sheath. So the initiation is going to happen at the axon hillock, and then gaps called nodes in the myelin sheath are going to allow saltatory conduction of the action potential. Saltatory means to jump. So in green, I'm coloring these nodes of RON-VA. That's where we're going to see a propagation. And each node has a concentration of those voltage-gated sodium and potassium channels so that when they open, you can get a depolarization just in that patch of the membrane. This word saltar means to jump. So first you have threshold reached and an action potential initiated at the axon hillock, and then it jumps, boom, 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 all the way to the end. And at each one of those patches of membrane, you have to once again depolarize and repolarize that patch of membrane. So notice in the little drawings I've made after that initial one, you don't have to worry about threshold anymore. It just immediately depolarizes up and then continues on. And that's why we say an action potential is all or nothing. Because if it gets generated and initiated at the axon hillock and there's enough myelin along the axon, it should make it all the way to the terminal. And then what's waiting at the end? Well, you could have a dendrite waiting, so another neuron. You could have a muscle fiber waiting, and then whatever it's released onto is going to cause a muscle cell to contract. Or it could be a gland, and maybe it will make sweat be released or saliva be produced or something like that. So in this picture, I'm drawing just the axon terminal and the target cell. In this case, I am going to make it a dendrite. How do you like the size of that dendrite that I drew? So I was talking to my 15-year-old about this, and he and I were discussing that it doesn't always have to be a neuron. That target cell could be the muscle cell or a gland cell. So we've got the cell body, and I'm making all those Nissl bodies, the rough ER in green, and then there's the axon hillock. Okay, so now looking back at the axon terminal, you have synaptic vesicles that contain neurotransmitters. And when the action potential arrives, it will open calcium channels on that axon terminal. Calcium is more concentrated outside of the cell, so calcium is going to be allowed to rush in, and when it enters, it will trigger those synaptic vesicles to be exocytosed from the axon terminal. So they're going to release their neurotransmitters across this gap called the synaptic cleft. So these neurotransmitters diffuse across, and then they bind to receptors on the postsynaptic membrane. I'm coloring the receptors in orange. I seem to always like to make them in orange. And then the neurotransmitters are binding to those receptors, and they're going to make something happen. So it's now gone from this electrical action potential to a transmission that's chemical. So we call this a chemical synapse. Now the receptor binding will open ion channels. It depends on what the neurotransmitter is and what kind of receptors the neurotransmitter binds to, what kind of ion channels will open. But here is a key concept. Those neurotransmitters are not going to enter the target cell. Only ions are going to get to enter. So try to keep that straight. And now I'm on this dendrite. I am drawing a bunch of ion channels. And these are going to allow ions to flow. Now remember, if it were sodium, it's going to flow in. If it's potassium, it would flow out. So in this picture, I'm using sodium as my example, and you can see that sodium rushing in, and it's going to change the membrane potential. If it is sodium, it's going to make it more positive. If it was a potassium channel that opened, the potassium will leave the cell, and it's going to make it more negative. Keep that in mind. If the stimulus is strong enough, it might be able to trigger an action potential at the axon hillock. So for example, if it's sodium that's coming in and it makes the membrane more positive way out there in the dendrite, if that signal can survive all the way to the axon hillock and is strong enough, then the axon hillock will fire an action potential. And I'm using this red line to show as the signal passes through the cell and makes it to the axon hillock, and then we'll make that axon hillock pink again. And there you have the potential drawn again. So I make the line where it goes up a little bit, and that represents, was the stimulus strong enough? And in this case, if it was, it will depolarize and then fire an action potential. So this whole picture, we're talking about how is the signal transmitted across this gap, this cleft between the two cells. In this one, I'm going to try to show you a little bit about how different neurotransmitters could have different effects at the synapse. So I'm going to draw side by side two cells as they're meeting up. There's two axon terminals, and then I'm drawing two dendrites again. It could be any target cell, but let's just say this is two dendrites, okay? So far they're the same, right? Okay, so far they're the same. They both have a nucleus, they both have the Nissl bodies, if they're neurons. And then they both have this axon hillock that I'm still coloring pink for you. Now the action potential arrives at the axon terminal, right? This is the same. And in the axon terminal, just like we looked at before, there are going to be synaptic vesicles that have neurotransmitters in them. Now on this picture on the left, I'm making the neurotransmitters yellow, like I did in the previous one. But in this other set of neurons that are communicating, I'm drawing purple neurotransmitters to indicate to you that this is a different kind of neurotransmitter. And that means it could have a different effect on the next cell. So what happens is that the action potential opens calcium channels. This is the same, right? And then the calcium can enter into the axon terminal. That calcium entry will trigger the exocytosis of the synaptic vesicles, and you can see them diffusing across the cleft. That part was fun to make. And then the neurotransmitters will bind to receptors on the postsynaptic membrane, and they will open channels on the postsynaptic membrane. So seriously, everything I just drew there is the same as what we did on the previous slide. It's like a review of the steps of synaptic transmission. But now I'm going to try to help you understand how different neurotransmitters could have different effects on their target cell. So if sodium or calcium channels are opened by the neurotransmitter, and I'll draw up here on the postsynaptic membrane sodium as an example, Do you see that as the sodium rushes in, it's making the whole membrane more positive? Because sodium is a positive ion, right? So you're bringing in plus charges, and it's making it very positive. So I'm just showing you sodium is coming in. And again, a reminder, notice that the neurotransmitter, the part I drew in gold, it is not entering, is it? It sticks to the receptors, and then it's the job of the sodium to come in the channels and make the change. So this is called an excitatory postsynaptic membrane potential, or an EPSP. And that's because excitatory, because it means it's going to end up being more likely to make the target cell fire an action potential. Now let's look at the other set of neurons and see that if potassium channels were the ones that were opened on the postsynaptic membrane, we have a very different thing happen. Potassium is always more concentrated inside of the cell, so voila, do you see it's going to go out? And as positive ions leave the neuron, well, what do you know? Relatively, it becomes more negative. So we represent that with little negative signs. This is an inhibitory postsynaptic potential, an IPSP. So it's going to inhibit the next neuron from firing an action potential. So again, looking at our positive, our excitatory one, the membrane potential becomes more positive, and it increases the likelihood of threshold being reached at the axon hillock, and therefore increases the likelihood of an action potential being fired. Whereas if the potassium channels open with an IPSP, the membrane potential becomes more negative. We even say that it's been hyperpolarized. It's like extra negative. And that means that we decrease the likelihood of an action potential being fired at the axon hillock. And this is the way that your brain can use different neurotransmitters to stimulate some activities and to inhibit others. And same with your muscles, to stimulate some muscles and then relax other muscles. So on this page, I just have an ultra quick summary of neurotransmitters, some of the different ones that you will hear about in your studies. Dopamine is associated with seeking reward and pleasure. It binds to dopaminergic receptors, and those start to die off in a disease known as Parkinson's. And so we know that dopamine is important in fine motor control as well, and not just in reward and highs. And then GABA, that's an acronym for a really long chemical name, but GABA binds to GABA A and GABA B receptors and is generally inhibitory. So like if someone takes benzodiazepines or alcohol, then they can stimulate GABA and it causes their brain to just kind of quiet, right? So they're going to be less anxious, and that's why people sometimes seek out those kind of medications and drugs, because they're trying to calm their brain. Maybe not the healthiest way to do it, but they're trying to do that. And then glutamate, this one will bind to receptors known as NMDA and is very, very, very common in the brain and is a big part of how we learn and remember and build pathways in our brain. Serotonin is important in stability of mood and binds to so many different kinds of what are called 5-HT receptors. And this one's also very active in our gut, I should have mentioned. There's a lot of neurotransmitters in your gut as well, not just serotonin. Then acetylcholine, otherwise known as ACH. It's very important at the neuromuscular junction as well as in your brain. And it is the mediator of rest and digest. So it has a lot of roles. And it will bind to cholinergic receptors. There's nicotinic, those are like the ones on your neuromuscular junction to make your muscles contract. And then there's muscarinic, which are part of your parasympathetic nervous system for the most part. And then norepinephrine, this one will bind to adrenergic receptors. There's different kinds of those. And norepinephrine is known for mediating your fight or flight response, and also important in attention, concentration, your mood, things like that in your brain. And then I added endorphins, and I'm sure that neuro people will be annoyed by this, but it's because it's not technically a neurotransmitter, but everyone's heard of opioids and endorphins. So it's really similar, and they do bind to receptors in the brain. They're just, I think they're like a bigger molecule than neurotransmitters, and they're associated with all kinds of pain blocking and helping you to feel good. So if you've heard of like the runner's high, it's oftentimes a release of endorphins. Or if you were to break your leg and be in a lot of pain, your body will instinctively release endorphins to try and blunt that pain for you, to get you through it. Okay, we did it. So that was our intro to the nervous tissue. I hope you enjoyed this video. I will attach a link to the PDF in the description below so that you can get a PDF of all the slides. I hope you have a great rest of your day. For me, it's nighttime here in Alabama, and I will see you in the next video.